Introduction. Welcome to today's lecture on advanced topics in hormones and liver function. In this comprehensive lecture, we will explore various aspects of hormonal regulation in the liver, focusing on the impact of hormone signaling pathways on liver gene expression, the role of liver hormones in regulating lipid droplet metabolism, and the hepatic effects of sex hormone binding globulin, SHBG. We will also delve into the hormonal regulation of hepatic drug metabolism and detoxification, thyroid hormone-mediated regulation of liver mitochondria function, and the influence of adipose-derived hormones on liver inflammation and fibrosis. Furthermore, we will dive into the involvement of liver hormones in the regulation of energy homeostasis, the disruption of liver hormone signaling and insulin resistance, and the crucial role of glucocorticoid receptors in hepatic gluconeogenesis and lipogenesis. Additionally, we will explore the hepatic effects of growth hormone-releasing hormone and somatostatin, as well as the impact of liver hormones on hepatic stellate cell activation and fibrogenesis. By the end of this lecture, you will have gained a comprehensive understanding of the intricate relationship between hormones and liver function. So, let us now begin our exploration into these fascinating topics. The liver plays a vital role in maintaining homeostasis by regulating gene expression in response to various hormonal signals. Several hormone signaling pathways, including insulin, glucagon, and growth hormone, have profound effects on liver gene expression. For instance, insulin promotes the uptake and storage of glucose and fatty acids, while glucagon stimulates the breakdown of glycogen and lipid degradation. Growth hormone, on the other hand, supports hepatocyte growth and proliferation. These hormones, through their respective signaling pathways, modulate the expression of genes involved in glucose metabolism, lipid synthesis, and cell growth to maintain systemic metabolic balance. Lipid droplets are essential organelles involved in the storage and mobilization of neutral lipids in the liver. Several liver hormones, such as insulin, glucagon, and adiponectin, play pivotal roles in regulating lipid droplet metabolism. Insulin promotes lipid storage by activating lipogenic enzymes and inhibiting lipolysis. Conversely, glucagon stimulates lipolysis and fatty acid oxidation. Adiponectin, secreted by adipose tissue, enhances insulin sensitivity and inhibits hepatic lipogenesis. Understanding the intricate interplay between these hormones and their effects on lipid droplet metabolism is crucial for comprehending liver lipid homeostasis. Sex hormone binding globulin, SHBG, primarily synthesized in the liver, regulates the bioavailability and distribution of sex hormones such as testosterone and estrogen. Besides its role in hormone transport, recent studies have revealed other hepatic effects of SHBG. For instance, SHBG has been shown to have anti-inflammatory properties in the liver and may play a role in modulating glucose and lipid metabolism. Understanding the implications of SHBG in hepatic physiology and its potential therapeutic applications is an area of active research. The liver plays a crucial role in drug metabolism and detoxification, and hormone signaling pathways can significantly influence these processes. Several hormones, including glucocorticoids, thyroid hormones, and sex hormones, have been shown to regulate the expression and activity of drug metabolizing enzymes and transporters in the liver. For example, glucocorticoids induce the expression of cytochrome P450 enzymes involved in drug metabolism, while thyroid hormones modulate the activity of drug transporters. A comprehensive understanding of the hormonal regulation of hepatic drug metabolism is essential for optimizing drug therapies and minimizing adverse effects. Thyroid hormones play a significant role in regulating liver metabolism and energy expenditure. These hormones are involved in the regulation of mitochondrial biogenesis and function in hepatocytes. Thyroid hormones stimulate the expression of genes encoding mitochondrial proteins and enhance oxidative phosphorylation, leading to increased ATP production. Moreover, they influence the balance between fatty acid oxidation and lipogenesis in the liver. An in-depth understanding of the thyroid hormone-mediated regulation of liver mitochondria function is crucial for deciphering the metabolic alterations associated with thyroid disorders and exploring potential therapeutic interventions. Adipose tissue secretes a variety of hormones, collectively referred to as adipokines, which have multifaceted effects on liver physiology. Adiponectin, for instance, exhibits anti-inflammatory properties and plays a protective role in the liver by suppressing hepatic inflammation and fibrosis. Conversely, other adipokines like leptin and resistin can promote liver inflammation and fibrosis. The intricate interplay between adipose-derived hormones and liver inflammation and fibrosis is an area of active research, as it holds promise for developing novel therapeutic strategies to combat liver diseases. Maintaining energy homeostasis is crucial for overall health, and the liver plays a critical role in this process. Several liver hormones, including insulin, glucagon, and growth hormone, are key regulators of energy balance. These hormones act on various tissues, including the liver, to coordinate glucose and lipid metabolism, promoting energy storage or mobilization as per the body's needs. Understanding the complex hormonal regulation of energy homeostasis is fundamental for devising interventions to combat metabolic disorders like obesity and diabetes. Insulin resistance, a hallmark of type 2 diabetes, is characterized by impaired insulin signaling and dysregulated glucose metabolism. The liver plays a central role in the development of insulin resistance. In this topic, we will explore the mechanisms through which insulin resistance disrupts liver hormone signaling, leading to aberrant glucose metabolism, increased hepatic gluconeogenesis, and dyslipidemia. A detailed understanding of these mechanisms is crucial for developing targeted therapies to improve insulin sensitivity and manage diabetes-related complications. Glucocorticoids, primarily cortisol, have diverse effects on liver metabolism. They influence gluconeogenesis, lipogenesis, and insulin sensitivity, among other processes. Glucocorticoids exert their actions by binding to glucocorticoid receptors, which regulate gene expression and modulate various metabolic pathways. In this topic, we will delve into the role of glucocorticoid receptors in hepatic gluconeogenesis and lipogenesis, shedding light on the intricate regulatory mechanisms controlling glucose and lipid metabolism in the liver. Growth hormone-releasing hormone, GHRH, and somatostatin, two peptide hormones secreted by the hypothalamus, play crucial roles in the regulation of growth hormone, GH, secretion. GH, in turn, exerts diverse effects on liver physiology. In this topic, we will explore the hepatic effects of GHRH and somatostatin, including their influence on liver growth, metabolism, and insulin sensitivity. Understanding the interplay between these hypothalamic hormones and liver function offers insights into the regulation of growth and metabolism essential for normal physiological development. Hepatic stellate cells, HSCs, are key players in liver fibrosis, a pathological condition characterized by excessive accumulation of extracellular matrix proteins. Liver hormones, such as insulin-like growth factor 1, IGF-1, transforming growth factor beta, TGF-beta, and leptin, have been implicated in the activation of HSCs and promotion of fibrogenesis. In this topic, we will explore the impact of liver hormones on HSC activation and fibrogenesis, highlighting the potential therapeutic implications of targeting hormone signaling pathways in liver fibrosis. In conclusion, the liver is a complex organ intricately regulated by various hormones. Through this comprehensive lecture, we have explored the impact of hormone signaling pathways on liver gene expression, the role of liver hormones in regulating lipid droplet metabolism, and the hepatic effects of sex hormone binding globulin, SHBG. We have also discussed hormonal regulation of hepatic drug metabolism, the influence of adipose-derived hormones on liver inflammation and fibrosis, and the involvement of liver hormones in energy homeostasis. Furthermore, we have investigated the disruption of liver hormone signaling and insulin resistance, the role of glucocorticoid receptors in hepatic gluconeogenesis and lipogenesis, and the hepatic effects of growth hormone-releasing hormone and somatostatin. Lastly, we have explored the impact of liver hormones on hepatic stellate cell activation and fibrogenesis. Through a detailed examination of these topics, we have gained valuable insights into the intricate relationship between hormones and liver function. This knowledge will undoubtedly aid in improving our understanding of various hepatic disorders and in developing innovative therapeutic strategies to combat liver diseases.